Yes, good evening to everyone. I'll present the screen and you tell me if you can see the PPT. Okay. Able to see. Okay. So, hello everyone, welcome back. This is a revision class. Okay. So, the last week class, that is week 12, this we will discuss day after tomorrow, that is on Friday. So, today is your revision class. So, in this class, it is going to be a rapid fire round of questions. And welcome back after Navratri, after Durga Puja. I hope you had good holidays. Okay, so we'll go through everything quickly, all the questions, and then these are all. So the initial part will be your practice questions, and then after that there are questions from CSIR Net, from GATE BT and GATE Excel. Okay, all the answers are also there. All the websites from which I have taken the questions and the answers are provided at the reference section. Okay, so you can go through that and you can practice the rest of the questions. So I've selected only the questions that are relevant here, but I would encourage you to look up and go through all the previous year's questions. All right. Okay, so let's begin. Right, so in today's class, we will not be discussing the topics as such. We will only be solving the questions. If you're having any doubts, you can ask me. Okay, and if there are any more doubts, then you can always look up and find for yourself. Okay, so we'll start. Right, so these are MCQs and MSQs for your practice. Now we will start with the first question. These kind of questions we had practiced very well, right? During our, I think week 2 or week 1. So you should be able to solve these. So here they are asking you the PI of this particular tripeptide. This tripeptide is made up of 3 amino acids. All the pKa values are given to you and they are asking you to solve this. Okay, it's a simple question, but this is a very scoring question. These kind of questions will come from your exams. So it's very important that you know how to solve them. So you can solve them and you can answer in the chat box. Okay, and in the next slide, we will look at the answer. It's a simple question. You should be able to solve this easily. So we know that for PI, the two pKa values that are closest to the pH at which the net charge is going to be 0, those two are taken into consideration and the average of those two are taken as PI. So you tell me from here which two pKa values are going to be most important in rendering 0 net charge to the tripeptide. Okay, shall we go and look at the answer then? Let's look at the answer, okay? So see, what is happening? You know all these 4 pKa values are given. right? So when the pH is less than the first pKa value, then your net charge is going to be +1. As we increase the pH, the net charge is going to become 0. Then the net charge is going to become -1, then -2, and then it is going to become -3. So the two pKa values between which the net charge is going to be 0 are this and this. And so therefore your Pi is going to be 3.03. That is how we do it. If you go back to our previous classes, you will see that that is how we had calculated earlier too. Okay. So if you have forgotten a little bit, it's okay. You can always go back and you can always brush up the concepts. Right. Now we will go to two more questions. You can try to answer these two questions in the chat box. So the first question is, what technique is suitable for investigating the kinetics of protein folding and unfolding transitions? So different biophysical techniques are given. Which techniques do you think would be most suitable to study protein folding and unfolding? Okay, so circular dichroism spectroscopy would be very important to know about the secondary structure. So what happens to the secondary structure when the protein is folded and unfolded? That you know, right? So what is going to be most suitable technique to study protein folding and unfolding? You know about X-ray crystallography, you know about FRET, you can make a guess. And then we have which method is most suitable for determining the stoichiometry of the protein complex. So stoichiometry means what number of exactly what subunit is involved in formation of the functional protein, right? So that you can know from some of these techniques. So which one do you think would be most appropriate? There can be more than one answers. Okay, it is possibly an MSQ. So you can see which one you can use. For stoichiometry, which all would you use? Okay, let us look at the answer. All right. Right. So see, for this one, you can see that differential scanning calorimetry would be very appropriate to study the kinetics of protein folding and unfolding. For stoichiometry of protein complexes, I would say analytical ultracentrifuge, size exclusion chromatography, these two would also be very efficient in studying the stoichiometry of protein complexes. Okay, so you can look up. Right, now we go to another question. This is also a simple question, again on your biophysical techniques. So which one do you think is going? This is a match the following question. All right. So which type of matrix is used in which kind of chromatography? This kind of question is also asked in your exams. Okay. So you should be well versed with this. So for affinity chromatography, you know that NINTA is used. So then which one is going to be? See, I gave you one clue. Now it should be easy for you to answer. Are we getting any answers in the chat box? Okay, we have one answer and that is C. Alright, let's look at the right answer. Yes, that is correct. C is the right answer. So affinity chromatography goes with NINTA matrix. Gel filtration chromatography goes with your what? This one, biogel P. And then cation exchange means that the resin is going to be negatively charged. So then carboxymethyl cellulose would have the carboxyl group. Carboxyl group is going to be negatively charged, right? So it is going to be 2. And hydrophobic interaction chromatography, this is going to have 1. That is your silica gel. All right. So C is the right answer. Now we have one more interesting thing. Which of these amino acids are glucogenic and which of these amino acids are ketogenic? Now this is something interesting because you might have heard about this keto diet. Everybody is going for these fasting, intermittent fasting, and then keto diet and everything. So what do you think is these ketogenic amino acids and glucogenic amino acids? So we will actually read about this. In case you are not aware of that, we have the next upcoming slide on ketogenic and glucogenic amino acids. So these kind of questions, if you're attempting, let's say DBT, if you're attempting CSIR, then these kind of questions can come in your section B of CSIR. Okay, and for DBT also these kind of questions can come because DBT always asks very direct questions. Glutamine. So you're saying that which all amino acids are considered glucogenic. Okay. All right. So the thing is, which is considered glucogenic? I think this the question should be in this way, which is not considered glucogenic, because glutamine and alanine, all of these, alanine, aspartate, these are all basically glucogenic. So in that case, the answer should be in this way, which is not considered glucogenic. Then you would select your leucine. Okay, so I will change this. Right, we have changed this, and now you know that these three are right answers, and we will talk about so right, okay, now we will talk about these ketogenic and glucogenic amino acids. All right, so see, the amino acids that can be broken down to acetyl coenzyme A are used in the citric acid cycle, but then mammals cannot synthesize glucose from acetyl coenzyme A. Alright, this fact is the source of the distinction between glucogenic and ketogenic amino acids. So some amino acids are broken down into glucose. So they are used to form glucose. Some amino acids are used to form ketone bodies. That is what makes them glucogenic and ketogenic amino acids. A lot of amino acids can be used in both the pathways. So they are both glucogenic and ketogenic. Alright. So glucogenic amino acids can be converted into glucose with oxaloacetate as the intermediate. But ketogenic amino acids cannot be converted into glucose. Now we will look at the pathway. You can go through these differences. Okay, this table is given for your ease, and we will go to this particular chart. This chart tells you very clearly which all are glucogenic amino acids, which all are ketogenic amino acids. So the glucogenic amino acids are shown in red, ketogenic amino acids are shown in green, and the amino acids that are both ketogenic and glucogenic are shown in violet. Okay, so you can see that initially this is your glycolysis, and after glycolysis it is entering into the site, what is that? Your TCA, right? So that is your Krebs cycle. It is entering into your Krebs cycle, and from there all of these different amino acids are involved in forming different kinds of products in the Krebs cycle, right? That is how the amino acids can function as glucogenic and ketogenic amino acids. Okay, now we'll move on to the next question. Right. So this is a question. This is a relatively tough question. So you can read this and you can try to answer. Okay. Many concepts are clubbed into the same question. I think this is the last question for your practice, and then just right after we have CSIR net previous year's questions coming up. We have GATE BT and GATE Excel previous year's questions coming up as well. Okay, so try to answer this question. So they are telling you that the folding and stability of a protein structure are influenced by various interactions and modifications. Consider a hypothetical protein, protein X, which has 200 amino acids. The following information is available. So protein X has two domains. Domain A is made of 100 amino acids. Domain B is made of again 100 amino acids. Domain A is an alpha helix. Domain B is a beta sheet. Okay, and you see that domain X undergoes a post-translational modification where phosphoryl group is getting added on the serine residues. Okay. And in the domain B, there is lysine, and on the lysine, acetyl group is getting added. Right? And then you have glycosylation. So what is glycosylation? Sugar moieties. Sugar moieties are getting added onto asparagine residues, that is N glycosylation, right? In the asparagine side chain to N to nitrogen, this entire sugar group gets added, that is N glycosylation. But if it is added to O, then it is called O-linked glycosylation. Okay, right. Now the protein is found to denature completely when the pH is dropped below 5, but retains partial structure at pH 6 due to the persistence of ionic interactions involving charge residue in both the domains. Now we will look at the question. This is what they tell you at first. Well, all right. Now, okay, I think it is already marked. Okay. So since this is already marked, we will just go through it. Okay. All right. So the phosphorylation, now they are telling you which is most accurate regarding protein X. And then we will look at the explanation why that is most accurate. Okay. So the first option is phosphorylation of the serine residues in domain A enhances the alpha helical stability by increasing the intrahelix hydrogen bonding. The second option is the acetylation of lysine residues in B is likely to weaken the ionic interactions within the beta sheet structure by reducing the positive charge. So lysine is supposed to have positive charge. It is supposed to form hydrogen bond with another beta strand, right? But then when you are adding this acetyl group means it is obviously CH3CO. So it is having this carbonyl carbon and it is having this CO, CH3CO, it is negatively charged. So then what is happening? The positive charge is getting weakened and therefore the hydrogen bond is getting affected. But they are saying that, yeah, so then that is definitely more accurate. Then you have disulfide bridges in domain B are most likely to break at pH 6 due to increased competition from the protonated cysteine residues. Okay, we will look at that in the next slide. Glycosylation of asparagine residue in domain A increases the flexibility of the alpha helix by disrupting the hydrophobic interactions. Now glycosylation, you know that this is a huge group. Every glycosylation, there are many small sugar moieties involved, right? If such a huge group is going to get added to your protein, it is in no way making it flexible. So this cannot be true. It cannot increase the flexibility of the alpha helix. Okay, that is not possible. So we will look at the explanation. Right. So here we have it. This is your option A. Option A is incorrect because phosphorylation typically introduces negative charge to serine residue, which can disrupt rather than enhance the intrahelical hydrogen bonding. Okay. Then you have option B. Option B is correct because acetylation neutralizes the positive charge on the lysine residue, which would weaken any ionic interactions with negatively charged side chain or phosphate group that we talked about, right? So hydrogen bonds are getting disrupted because of this oppositely charged group added. Fine. C is incorrect. Why is C incorrect? Because your disulfide bond is most stable between pH 3 and 8. Here they are saying that at pH 6 the disulfide bond is broken, but that is not possible. Correct. So disulfide bond is most stable between pH 3 and 8 and it can break at a pH of around 10 to 12. So here they are mentioning critical pH of 10.5. Okay. Then you have D, option D. So option D is incorrect. Why? Because glycosylation increases the structural stability. It is making the entire structure more rigid, right? It is not increasing the flexibility. It is definitely decreasing the flexibility. So that is all about this question. Okay, we'll move on to the next question. Here are your previous year's questions. Okay, you can try to solve this question. This question is taken from CSIR NET 2023. Okay. Now, one very important thing about solving previous year's questions is once you start with the previous year's questions, you will feel like you know nothing. Okay, everything will seem very difficult and everything will seem so much beyond whatever you have studied. Then what you will do is, please don't let the book be and go back to your studies. Don't do that. That would be a mistake. Try solving the questions. Try looking up answers. If you are learning anything new, that is a good thing. You write it down. Take a different notebook. Write down everything that you're learning new. Once you start solving at least 2 papers, 3 papers, once you have solved, you will realize that most of the questions, most of the concepts are repeated. And once you understand how to solve a particular question, it is going to help you in the next papers as well, next few papers as well. Okay, so concepts are always repeated. Solving more is going to help you to understand the weightage of each concept. It is going to help you to understand how to approach a question. So if you think you don't know something, learn it from the question and its explanation. Do not go back to your books right away, leaving the question back. Don't do that. Okay. Right. So try to solve this question now. So a polypeptide was subjected to the following treatments with the indicated results. First, there is acid hydrolysis. Okay. And here are the different results that they are getting. Then there is aminopeptidase M. So they are not getting any fragments. Now, if you think what is aminopeptidase M doing, right? What is that doing? What is this doing? Then you will obviously read up. What is aminopeptidase M doing? It is cutting at the what? Aminopeptidase. So it will definitely cut at the N terminal. So that you read. Okay. But then it is not generating any fragments. Why is that so? What is the specificity of this aminopeptidase? Then you have carboxypeptidase A and B. What is the difference between carboxypeptidase A and B? We have that in the next slide. So we will look at that. Okay. Carboxypeptidase means it is going to cleave at the C terminal. Okay. And then we have trypsin digestion followed by Edman degradation of the separated products. Now, if you remember, I had given you a list of the favorable proteolytic cleavage site, and I had told you that every single year you are going to get questions from these. One second, I'll be back. Okay, just give me a minute, please. Just give me a minute, I'll be back. Let's continue. So here, okay, let's continue. All right. Okay, so trypsin. Now you know where trypsin cuts. Trypsin cuts at HRK, right? 3 amino acids, and there should not be proline on the right hand side. So that is where trypsin cuts. Where does chymotrypsin cut? Where does your pepsin cut? Where does, there are so many different enzymes, lipases, etc. So where do these enzymes cut? So the proteolytic cleavage is a very important question. Okay. And then what is Edman degradation? Edman degradation, I'm sure you know. Okay. So go through it. If you've forgotten, it's fine. It is always very important to solve questions because that will make you realize where you stand. Okay. And how much needs to be revised. It's totally fine if you don't remember something. It's always okay. Now, after this trypsin digestion, Edman digestion has been performed and they are getting these products were separated. Fine. So, which one of the following represents the primary structure of the peptide? Okay. So, to solve that, you need to know the function of all of these different enzymes that were used in the hydrolysis process or proteolysis reaction. Okay. So, the different primary structures are shown here. So, you can answer in the chat box. Otherwise, we'll go and look up. We'll look at the answer. This has been taken from CSIR NET 2023. Okay, this kind of question you can expect in section C. Section C is always analytical questions, and your concepts need to be very clear so that you can answer the section C questions. Now another important thing is that there will be 75 questions. From there you only have to attempt I think 25. So if you are not able to answer most of the questions, please don't feel disheartened. You only need to answer few questions to clear it. But those few questions you have to answer properly. Okay. So whatever you are studying, make sure you are very well versed with that. You don't have to study all the topics, everything. Whatever you are studying, study it properly. Okay. We'll look at the answer for this question then. Okay. We'll look at this one. See. So which one of the following represents? If you are not able to see the PPT, please tell me. Okay. Alright, so carboxypeptidases and aminopeptidases are exopeptidases that remove the terminal amino acid residue from the C and N terminal respectively. Carboxypeptidase A cleaves the C-terminal peptide bond of all amino acids except proline, lysine and arginine. Carboxypeptidase B is effective only when arginine or lysine is there at the C terminal. So these two are basically more or less antagonistic to each other's function. Okay. Now carboxypeptidase C on the C terminal cuts on any residue. Absolutely any residue. Okay. They have used carboxypeptidase A and B. Only A and B. Okay. Aminopeptidases are catalyzing the cleavage of amino acids from the amino terminus of the protein. Amino terminus M catalyzes the cleavage of all free N-terminal residues. Okay. Rest, trypsin, it's there in our previous slides. Please go back and read. Edman degradation, you know Edman degradation. It is possibly there in our slides. If it is not there, you can always read it in Boyt and Boyt or Cleninger's biochemistry. Okay. From there you will realize that this is the cyclic peptide which is actually the structure. So once you go through it, you will realize that this is the cyclic peptide that you are getting. Okay. Alright. Now we will look at the next one. So next is a question which is taken from CSIR Net 2023 once again. I told you that antibiotics are very important. You will always get questions from antibiotics. So try to see if you can solve any one of these. Okay. So which one of the following statements is correct in the context of erythromycin mediated inhibition of protein synthesis in bacteria? What does erythromycin do in bacteria? Mode of action of the antibiotics and class of the antibiotics. These two you must remember. Antibiotics are possibly having 5 different classes. 2. Okay, so you're saying that 2 is the right answer. Erythromycin inhibits protein synthesis at the step of elongation and it prevents formation of even the first peptide bond. Okay, let's see. Let's see which is the right answer. Right, so the right answer is actually 1. Okay, so erythromycin binds to the 50S ribosomal subunit and inhibits the peptide chain elongation. So it nonetheless allows translation of the first few amino acids. Okay, so you have to read the statements properly and see these kind of things. Unless you practice, you cannot ever master, right? So in the book, only that tiny bit will be given that it prevents elongation. But then when you go through the previous year's questions, you will realize what CSIR Net expects you to answer. Okay, what is accepted as the answer. Then you will realize that, okay, it allows first few residues and then it stops. First few codons, sorry. Right, so it is very important to practice and practice. There are some more amino acids given here. For example, chloramphenicol, which also binds to 50S. You can go through it and see what it does. It inhibits the peptidyl transferase enzyme. Right. So just prevents protein synthesis is the broader function. But then exactly what happens that you need to know, because that will come as match the following. That is very important. Please know it. Okay. Right. Now we go to the next one. These kind of questions come in, I think, DVT, GATE, and not sure about ICMR, but yeah, DVT and GATE, you can expect these kind of questions. Okay, we have one more question on these protein cleavage. So protein cleavage, see in CSIR net 2023 only, there are already 2 questions. We already got 2 questions here. So you can try to answer this one also. So that's what, if you know one topic properly, and if it is an important topic, you can expect minimum 2 questions from that topic. And this is from section C, so it carries a lot of marks. Okay, so if it is correct, you score a lot of marks already. Okay, now see, the following statements are made about digestion of proteins and enzymes involved. So option A is chymotrypsin does not generate peptides with the C-terminal neutral amino acids. If you remember chymotrypsin, you will be able to answer this. Trypsin generates peptides with C-terminal basic amino acids. Okay. Now you have to remember where trypsin cleaves, whether it is the C-terminal of basic amino acids, whether it is the N-terminal of basic amino acids. Okay. Right. Carboxypeptidase B acts on aromatic amino acids. We talked about carboxypeptidase B just now. So you know which is not correct. Now see, carboxypeptidase A employs zinc for hydrolysis. This is why we had this metal ions and all these enzymes that are important, that require metals for their activity. Right, so it's important to know that too. Now the brush border enterokinase has no polysaccharides attached to it. For this you will have to know physiology. Okay, what is there in your intestine and digestive system. So when you study that, you will possibly remember this. The final digestion of amino acids occur in the intestinal lumen, the brush border and the cytoplasm of the mucosal cells. So which of these are, all of the combinations are correct. So let's try to solve this. 4. So you're saying that 4 has all of these correct. Okay, let's see. Yes, very good. 4 has all of these correct. See. So carboxypeptidases, and now here this table is given. Please remember this table. It is important. CSIR net itself basically gives you this table. So it's very important that you remember this. Alright. Now see, carboxypeptidases and aminopeptidases are exopeptidases. So that means they are going to cleave between the first and second amino acids on C and N terminal respectively, right? And carboxypeptidase A cleaves the C terminal peptide bond of all amino acids except proline, arginine and lysine. B cleaves only arginine and lysine, and C cleaves all, right? Aminopeptidase catalyzes the cleavage of all amino acids from the N terminal, and M goes for all free N terminal amino acids, and then you will realize which of the statements are wrong then. Okay, and then you can be able to solve. Okay, right. Now we go to the next question. Okay, this is a question on enzyme. So it's a question on enzymes. Once again, CSIR net 2023. So enzyme properties are given and the formula is given or expression. Okay, we had learnt about this in our previous lectures. So enzyme specificity. Which one do you think? If you remember the definition, you will be able to solve this. Michaelis constant. This should be simpler for you. See, this you must know. This all of you know, I'm sure you know. Yes, C is 2, correct. So that means either 3 is correct or 4 is correct. Okay. And then you have turnover number and you have catalytic efficiency. So then which is the right answer? Okay, we'll see. We'll see the right answer. We have got the answers to. So the right answer is 3. Did you all answer 3? Yes, very good. So you all answered 3. Very good. Yes, if you remember the formula, it will be easier. Now, based on this formula, there are going to be numericals. There are going to be NAT in GATE exam. There are going to be questions in your DPT exam, CSIR exam. So, please remember the formulae and please practice, okay, the numericals. So, the specific activity of the enzyme is amount of enzyme activity per unit of protein. That is enzyme in mg, and it is a measure of the enzyme's catalytic efficiency, and it is often used to assess the purity of the enzyme preparation. Specific activity is calculated by dividing the total enzyme activity by the amount of the protein present in the sample. Provides a measure of the concentration of active enzyme molecules in relation to the total protein content. Okay, so please try to go through our previous slides. Everything is going to be given to you there. Okay. Fine. So we have the next one. Next one is also once again CSIR net 2023. Try to solve this. In this question, you should be able to match at least two, because we have talked about two in our class, right? So one is mass spectrometry and the other one is isoelectric focusing. You know these two. These two you must be able to match. If you can match these two, it's all done. You can solve the question. Try. Mass spectrometry, what is it used for? And isoelectric focusing. If you remember, mass spectrometry is used for knowing the exact sequence of residues in a protein. What else can it tell you? Isoelectric focusing means that you are focusing all the proteins at a specific pH. Okay, we will look at the answer in case you are confused. The right answer is A. That is because, okay, no, it's not 4. It is 1. Why is it 1? Because mass spectrometry is used also to identify the post-translational modifications in a protein. The second thing is this PYQ, it's a DNA sequencing method. Okay, so PYQ is a DNA sequencing method and it is helping you in your single molecule real-time sequencing. So if you study about this, I think that is a third generation DNA sequencing, if I'm not wrong. You can read about it. And then we have, SMRT, which is single molecule real-time sequencing, right? SMRT goes with PYQ. And you have isoelectric focusing. So if you remember 2D gel electrophoresis, and the first step is your isoelectric focusing. The second step is SDS page. So isoelectric focusing means all the proteins carrying the same PI, all the proteins with the same PI will come and precipitate at a specific point. That can be used to separate isoenzymes. What are isoenzymes? They are very similar. They are having very similar properties, right? So you can separate them. Even the slightest differences in the PI can be separated using isoelectric focusing. Pulse field gel electrophoresis. In this, what happens? This is used for separation of whole chromosome. So what is happening? You are providing a pulse and there is movement along the gel and you're stopping it, and then again you're providing a pulse and then again there is movement. So this is how you can separate whole chromosomes. Fine. We'll go to the next question. Try to solve these two. These are GATE Biotechnology 2019 questions. We don't have too many questions today. We just have a few more and then that's all. We only have a few more questions. So see, these are also very logical, very simple questions. All you need to do is just understand everything properly. Okay. So hexapeptide P has an isoelectric point Pi of 6.9. Hexapeptide Q has a variant, it is a variant of P that contains valine instead of glutamate. So glutamate means it is going to be acidic, valine is not going to have any charge, right? And the two peptides were analyzed by Page at pH 8. Excuse me. So then what is going to happen in these two at pH 8? Which one is going to be negative? Which one is going to be positive? And which one is going to migrate faster? So if it is negative, obviously it will migrate towards the what? Anion goes to anode, right? So it is going to migrate to the anode faster. So now you tell me which one is going to be the right answer. People are leaving, people are bored. Okay, no problem. We'll solve the questions. If you're busy, it's okay, no problem. You can always look up the videos on YouTube and you can go through them. Chalo, try this. Okay, so you're saying that 1 is the right option. Alright. We go to the second question then. Second question, you have a match the following. So circular dichroism is important for what? Nuclear magnetic resonance is useful for what? This is also very simple. Try to solve this. So the question is, what is infrared spectroscopy useful for? First B and second for A. Okay, let's see. Second for A. Okay, right. We will look at the answer. Okay, so see, the first one is A. You guys are correct. Alright. And how is it A? We will go through that once. It is there in one of our previous slides. Why is it A? So if you see, this is how you will actually have to solve it. Okay. So if this is glutamate, then the pKa is going to be low. Right. And if the pKa is low, it is going to become negative quickly. If the pKa is not that low, it is going to carry positive charge. Right. And then what is going to happen at pH 8? That is what they have asked, right? So what is going to happen at pH 8? Then you can estimate. And then we will know which is going to be negatively charged and which is going to be positively charged. Okay, right? For this one, C is the right answer. Q and R, you can definitely match. Circular dichroism, you know that it is for determination of the secondary structure. Nuclear magnetic resonance is important for determination of the 3D structure. Infrared spectroscopy is important for identification of the functional group. Because, of course, you cannot estimate the molecular weight with infrared spectroscopy. You cannot do that, right? For that, you need what? SAC. So, yeah. And you also have analytical ultracentrifuge, which can be used for estimation of molecular weight. So then you have identification of functional group using infrared spectroscopy. Okay, we go to the next one. If you remember, we had talked about these antibody producing hybridoma cells. So we had talked about monoclonal antibodies, I think in our previous class or maybe one class before that. And I had told you that every single exam is going to ask you at least one question on this. So they are asking you how is this hybridoma cells getting formed? What cell types come together to form the hybridoma cell? That is one question. The other question is DNA polymerase in polymerase chain reaction. We know that DNA polymerase is having very high fidelity. What is fidelity? Fidelity means that it is going to be very less error prone. Right? It is not going to make mistake in incorporation of the nucleotide bases. How can you increase the fidelity? So what all factors affect its fidelity? That they are asking. For the first one, it's B. Yes. Okay, very good. What is it for the next one? What is the answer for the next question? You know about Mg2 plus concentration, you know that it is required. You know about annealing temperature. If the annealing temperature is very close to the melting temperature, right? So then what is going to happen? The specificity is going to increase. That means the only the what is going to happen? It's B again. Okay, let's see. P and R. All right, we'll see the right answer. Yes, so the first one, it is B, correct. B cell with a myeloma cell gives you the hybridoma cell. For the second question, it is PQ and R. So all of these are important. This is why solving questions is very important, because in the books they are never going to talk about pH, right? They will possibly not mention pH anywhere. Once you start solving the questions, you will realize that okay, maybe that is also important. All right. Now we will go to the next one. Right. So next one is again a question that looks very complicated, but it is a very simple question. You just read the question, you will be able to understand. Which of the following techniques cannot be used to determine the sequence of a novel protein? The first one is de novo sequencing by ESI-MSMS. So ESI-MSMS is tandem mass spectrometry, right? And then you have Edmond degradation. So in Edmond degradation, every single C. Yes, yes, correct. Why is it C? You tell me why is it C? Sure. You can speak. Tell us, Amisha, why is it C? So once you look at sequencing at first glance, you will be wondering which sequencing method should it be, and then you'll realize one is Sanger sequencing, right? So if you remember, Sanger sequencing is used for DNA, and all the rest of these are used for knowing the sequence of a peptide. So this is a simple question. All you need to do is just understand. Now the next question is something which is something very interesting. Okay, so they are asking you that a protein has 7 cysteine residues. The maximum number of disulfide bonds of different combinations that can possibly be formed by these 7 cysteine residues is. So this is a NAT, numerical aptitude test. This is from GATE Excel 2023. The one before this was GATE Excel 2019. Okay, so in 2023, this particular question was asked. Now the issue is that this question, the answer that they have given is different from the answer that is there on different websites. Okay, so we will look at the answer. For this one, you know that it is C. For this one, they have given the answer as 48. Okay, and they are only accepting 48 as the answer. Now, to understand how to solve this question, this is one place where they have explained how to solve this particular question. So they have used a specific formula, that is n factorial divided by r factorial into n minus r factorial. If you solve it according to this, then it becomes your 7 factorial divided by 2 factorial into 5 factorial. Correct? But the issue is that is going to give you 21 as the answer. It is not 48. Okay? It's going to give you 21 as the answer. Now if you are going to look at another way of solving this, this was given in Quora possibly. This was given in a different website. Okay? So if you follow this step, everything explanation is given. This is giving you 105 different combinations for 8 cysteine residues. So you can derive for 7 cysteine residues what should be the answer. Okay, that you can derive. But then you can look up whether this question was challenged or not, whether the answer was changed later on or not, because I have taken this question from IIT Kanpur's website, and there all the answer key and everything was given. Explanation was not given. So based on the answer key, this 48 is the answer, but you can always look up. So in case you are getting this question later on also, to understand how to solve this, I have provided you with 2 different solutions. This is one of the solutions through which you can do. Okay, and then you have the other solution, which is this. So you can go through these, all right, when you get time. Now we come to the last question of the day. Okay, this is the last question of the day. This is also taken from GATE Excel 2023. Please try to solve this. It is a simple question. Which of the following techniques can be used to separate proteins according to their molecular weight from a mixture of proteins? Based on their molecular weight. You should be able to solve this. It's a simple question. C. Okay. Yes, C is definitely one of the answers. We have more answers. Which all other methods can be used? B can be used. Yes. What else can be used? A, based on molecular weight. Ion exchange chromatography means based on your charge. Right? Based on charge differences. So no, ion exchange chromatography is not the answer. You tell me what all possible methods can be used. D is 1, C is 1. Can you use D? Think and say. Can you use D to separate proteins based on the molecular weight? Is it possible? Density gradient centrifugation, cesium chloride density gradient centrifugation, and sucrose density gradient centrifugation. These are also important to separate the proteins or any other components based on their molecular weight, right? So all B, C and D, they are all correct. This is question number 42 and you get 2 marks for this if you're correct. Alright. Okay. Thank you so much. These are your references. All the places from where I have collected all of these questions and the answers are all provided here. So you can go through them. Okay. And all the best for your exams. All the best for your life. And we will meet again day after tomorrow though. Okay. That will be your week 12. Thank you so much for your time. Okay, so are there any doubts, any queries, any questions? Otherwise, we can end the class. regarding the question about novel protein sequencing. Yes. What is the question? I'll go to that question then. On Friday, there will be any session? Yes, on Friday, you will have the week 12 session. This one. Yeah. So if you want me to discuss anything, how is Sanger sequencing? The answer. Sanger sequencing is for DNA sequencing, right? The rest of these are for protein sequencing. So they are asking you which cannot be used to determine the sequence of a novel protein. Sanger sequencing means it is going to be, it cannot be used. How pH affects PCR? You read about it. Okay, how pH affects PCR? Please read about it. So if you want to talk about any other topic, if you have not understood anything until now, I will recommend you to please mail me beforehand. Okay, I am giving you my mail ID and you can mail me anything that you have not understood, and then mail me at least 2 days prior. Then I can discuss with you on Friday. So you can mail me today or tomorrow. In the PPT also it is given that Sanger sequencing is used to sequence amino acids. No. Sanger sequencing is used to sequence DNA, right? Sanger sequencing and your chemical sequencing. These are for sequencing of DNA. How is Sanger sequencing done? Through DNA polymerase. So one chain is there and then you take another what? DNA polymerase and you add the DDNTP, right? Dideoxynucleotide. Then what happens? It will synthesize based on the complementary nucleotide, and wherever this DDNTP is there, it is going to stop there. And then you run all those on the gel and you will realize that at specific points these are all stopping, and then from there you can derive the entire sequence of the newly synthesized strand, and then you get the complementary strand, which is your parent strand, template strand, right? That is what is done in Sanger sequencing. For proteins, yeah. So, ma'am, can we expect MSQs in the final proctored examination? I am not sure about that. You can go through the NPTEL previous year's questions, okay, on the internet, and then from there you can see if MSQs are allowed. Okay. As far as I know, if there is any MSQ, they are going to ask you at the end of the options that A, only B, both A, B and C, that way it will come. So if it is an MSQ, it will be written there, tick all possible combinations and all, and it will be given that way. Both A, B and C, all of the above, that way it might come. Okay. Okay, so I'll give you my mail ID. If there is anything that you please repeat CSIR net 2023 today, first question of the session. All right, I will do that. Definitely I will. Give me a second. I will write down my mail ID. You can. So I am sending this to you. Any doubt, anything, please mail me by tomorrow so that I have some time to at least go through your mails and talk to you about anything that you're worried about. All right. Lecture materials of week 1, 2 and 4 are still not showing up. Is that so? In that case, I will either add the PPTs once again, or I will delete the present PPTs and then add once again into the folder. Okay. Then you can check, because I have added all the PPTs. Okay. We will go back to the first question of 2023 CSIR NET. Okay. Yes, this one. Tell me, what is your doubt? Okay, CSIR Net 2023, today first question of the session. Right. So see, the first question of the session, they are giving you a list of different type of things that are done to a specific protein sequence. Now you don't know the sequence of the protein, but you know what all can be done. Right, what all has been done that you know. So the first one is acid hydrolysis. So acid hydrolysis is going to tell you what all amino acids are there. So they have given that all of these different amino acids are there. You're like, okay, fine, great. Then we will go to the next treatment, that is aminopeptidase M. So aminopeptidase M can cleave any amino acid that is there on the extreme N terminal. Right, any sequence, it can be any nucleotide and it is just going to cleave that. Fine, then you get the fragment. Okay, then it is not giving you any fragments, right? So aminopeptidase M is not giving you any fragment, and that is an indication that there is no fragment on the, I mean, there it is not a linear peptide. How do you understand that it is not a linear peptide? Because both carboxypeptidase A and B are not giving you any fragment, plus aminopeptidase M is not giving you any fragment. These are all exopeptidases. So exopeptidases are only going to cleave the extreme end amino acids. If there is no extreme end in this peptide, it is not going to cleave anything. That is why in both step 2 and 3 you are not getting any amino acids. Now see, it would have been possible that you are giving carboxypeptidase A and B and it is not giving any fragments even if it is a linear peptide. When would that be possible? If the ends would have proline. If there would be prolines, right? So this C terminal, if there would be proline, only then it could be possible. Because A does not cleave when there is proline and B cleaves only when there are two residues and proline is not included. But then we are not seeing any proline in the first step, acid hydrolysis. So that rules out this entire possibility. Then you go to trypsin digestion. Trypsin is HRK. It cleaves. You get this specific fragment. You see that arginine is there at one end. You will know that the next fragment will start from this C terminal of this particular arginine. Then there is one lysine and you will see that okay, after lysine it was cleaved. From there you can understand, from the step 2 and 3 basically you can understand that it will not be a linear peptide, it will be a cyclic peptide, otherwise it would have been cleaved by the exopeptidases. Got me? Tell me if you have understood. Great. Okay, this is how you will be able to solve the questions. Once you start practicing the previous year's questions, you will understand so many different kinds of questions can come. And then you will basically think that way. Otherwise, at your first go, it will not come to your mind that, okay, this is also a possibility. Once you practice, practice and practice, you will realize that, okay, it is repeated. And one more thing that I'll tell you is questions are always repeated. In your NPTEL exams also, most of the questions are going to get repeated. Okay. At least the topics from which questions have been asked. Please go through those topics properly. Because people are going to ask similar questions only. Most of the questions, many questions actually, they are going to get repeated. Okay. So practicing is very important. And if you are having any issues with any answers to any questions, please get them clear. Alright. If you have any doubts regarding anything, you want me to explain any topic properly, once again, mail me in advance. Mail me today, mail me tomorrow. Alright. Day after tomorrow we have another class. So by tomorrow, please send me all the mails that you want me to talk about once again. All the best. Okay. Thank you so much. Thank you. If there are no more questions, we can end the session. Thank you so much. Okay, let's end the session then. Thank you to everyone.